How'd you do on this one? Okay, good. Okay. Because it's a big Okay. So Jason is recording today's review session. He hopes to post it to YouTube tonight, and we'll share the link as soon as possible. Thank you to Jason for doing that. Okay, now, if I ask, listen real quick, if I ask you the question with these options on exam 2, would there be one option or two options? Would there be two correct answers or one correct answer? Glutamine technically is a precursor to GABA because glutamine is needed to make glutamate. But I wouldn't do that to you. All right. Does it? Oh, okay. Good. Well prepared. That's okay. It's all good. Ten seconds. All right, let's see how you did. Let's see, inhibits NMDA receptors. Let's see if anybody was a smurf. Down. So far, everybody's bad. That's not a valid answer, but that's okay. I don't think bad is specific enough. Inhibits it. But there were other correct answers. Allows chloride to flow in, inhibiting. You guys see it's showing off. No.
Yeah, Brandon Ayuk's out. Braxton Berrios from Dolphins towards ACL. Oh, yeah. What? Oh, yeah. Yeah, it was a bad weekend.
All right, here we go. Click on the receptor that allows sodium to flow in. Look at that heat map. It's the AMPA receptor. Now, hold on. Why is that not the only correct answer? Okay, Priya's got it right. Because I told you, even through the NMDA receptor, it's mostly calcium, but there is some potassium and sodium. So again, there would have been two correct answers. That's why I'm doing these weird things on the in-class quizzes, just to probe, to make you I want you thinking about the whole system, not just one receptor. But I'm not surprised most of you picked AMPA, because sodium is its primary ion. Now this is where we get close. We get much closer to what exam 2 questions are going to look like, right? I don't want to go through everything today and read, but know your synthetic pathways really well. Know your synthetic pathways very, very well. 15 seconds. So I'm 99% sure I've secured a pediatrician who's a UK alum from Atlanta, Georgia, to come talk to this class on November 14th. Kid. I know, he's not a pediatric oncology or a neurologist. He's a general pediatrician. But he's also a donor. Right? We call him a rising donor. I had lunch with him last week. Rising donors, you want to make a name. And he really wants to talk to pre-med neighbors. All right. How'd we do? Awesome. Okay, now we're going to get a little more clinical correlated. Clinical correlates. Okay? What receptor system do we activate to help control seizure activities with medicines? What receptor system? I'm not talking about voltage-gated sodium channels, because there are medications to treat seizure disorders, migraines, and chronic pain disorders that block voltage-gated sodium channels. Ooh, what? I'm looking for a neurotransmitter receptor, probably. Is that big enough? You're going to have to answer this question in a different. I'm going to ask it in a different way on exam too. But so you need this. Why do you need to know about GABA uptake in astrocytes? Well, because one of the most commonly prescribed medications to treat seizure disorders, migraines, and chronic pain inhibits GABA uptake in astrocytes. So the answer here is GABA. Okay, that was a tough one. If I activate NMDA-type glutamate receptors, I'm going to induce seizures. I'm going to cause seizures. You didn't get past serotonin? Jason's still trying to catch up, folks. That's okay. All right. I have all of the slides out here. Shoot questions at me. What do you want to cover the most? What do you want to, is there specific things? Know your enzymes. Know those enzymes. Know the amino acid or the nutrient precursor. Is tryptophan, tyrosine, or choline the precursor to acetylcholine? It's an easy one. Okay? Know those enzymes, the precursors. We talked a lot about 5-HT2A receptors because of its unique signaling. Yeah. Yeah. Anybody have questions? We're going to do serotonin receptors. Anybody have questions about catecholamines? We really focus a lot on synthesis and metabolism. Okay? You don't need to know any structures. Know this pathway very well. I looked through exam 2 yesterday, and it's a lot of how is this made? What enzyme converts this to that? What's the amino acid precursor to dopamine and norepinephrine? Okay? Know your pathways. Nigrostriatal versus mesolimbic versus mesocortical. Know where neurotransmitters are made. For glutamate and GABA, it's easy because they're made everywhere. Know where serotonin is made, the raphe nuclei of the brainstem. Dopamine, ventral tegmental area of substantia nigra. Norepinephrine, locus coeruleus. For synthetic pathways, I 100% think the best way to do this, what do we do after catecholamines? Was that serotonin? Okay. 100% think for synthetic pathways, the best way for you to do this is to write out on a piece of paper and draw it out yourself. Don't do the chemical structures, right? But draw the enzyme, or the precursor, the enzyme into the next product. Serotonin, it's easy. All right, serotonin. Obviously know this. How does serotonin get in the brain? Know that. That's unique. So I will tell you, because we're doing enzymes and pathways, this exam does get a little bit harder, even than exam 1, which is actually potential like, neuroanatomy like, right? Which enzyme is part of the pathway that synthesizes both serotonin and the catecholamines? That's probably gonna be easy. Because they're structurally very similar. That's aromatic L-amino acid decarboxylase. Why are monoamines used to metabolize both serotonin and the catecholamines? Because they're structurally similar. They're not identical, but they're structurally similar. Okay?
Similar to both serotonin and the catecholamines?
Yes, structurally they're very similar, right? Again, don't know the B numbers. This is one way that Raphe nuclei are numbered. You don't need to know that here. Wait for med school or PA school or whatever. No chemistry, no structures. We really only talked about the 1A being the serotonin autoreceptor, right? The GI linked autoreceptor. We're not going to talk about it again for the rest of the semester. 2A, yeah, we're going to talk about it a lot when we do hallucinogens. This is that nasty GQ pathway. So yesterday in the graduate version of Intro to Neuroscience, I taught about endocannabinoids. Do you remember where endocannabinoid synthesis starts in relation to serotonin? 2 weeks, the grad students have to spit this back at me in essay form. Diacylglycerol, produced by the cleavage of PIP2, phospholipase C, when GQ is activated, chops PIP2 into IP3. Diacylglycerol, or DAG. Downstream, diacylglycerol produces endocannabinoids, the endogenous substances that activate cannabinoid receptors, just like THC from psychoactive hemp plants. That is probably also part of the runner high. Opiates and endocannabinoids. So when we talk about endocannabinoids, we're going to talk about GQ signaling. Okay? This is the same thing done in a slightly different form. But it's important. Important signaling stuff because it's a great model. What's unique about 5-HT3 receptors in the area postrema? It's a very important one in pharmacology. It's an inotropic. Is it anion or cation sensitive? Cation, calcium channel. What's the area postrema so important for? And serotonin 3 receptors. What? Ooh. Okay, I'm going to say blood-brain barrier. Is there one there? No. Who said what? Where? Chemosensory. Your vomit center. Yeah, blah, 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 blah. Okay. We did not go through serotonin syndrome other than to say it's bad. Okay, so don't worry about that. No. Okay. Don't know structure. I mean, you know what you need to know. Know everything about synthesis and metabolism. How glutamate and GABA are unique in terms of uptake after neurotransmission. Which part of the GABA metabolism mechanism could we activate with a pharmacological compound to control seizures? Questions? Specific questions? Now, these are two, the GABA and glutamate are tricky, right? Remember, glutamine is taken up by both the GABAergic neuron and the glutamatergic neuron. So no steps 1 through 6. No step 1 through 6. Yeah. Well, the astrocyte is the glia, the type of glia involved in glutamate and GABA uptake. Yeah, at that point. Now, a genetic, I want to see how you're thinking about this big picture. A genetic abnormality in which of these parts of the glutamate synthesis and metabolism pathway is a risk factor for seizure disorder. Okay. What would the abnormality be? Well, okay, so Priya's right. Genetic abnormality in the function, the DNA sequence of the excited amino acid transporter that sucks glutamate up is a risk factor for seizure disorders in a small number of people. Would that risk make the EAAT more or less active? Less active. So more glutamate is left in the synaptic cleft here to stimulate postsynaptic neurons. It's a small percentage of people, but we know if you have an identifiable risk, you're monitored much more closely for seizure disorders, okay? Big red X says it all. Esteban?
Regarding
Tropic.
glutamate receptors, we go over the function of canic acid.
No. Remember we talked, that's simply a Jeopardy level question. Canic acid is a toxin in red algae. It sometimes shuts down the Gulf beaches in Florida. Don't know the long chemical names, right? But do know how AMPA and NMDA receptors work together. How AMPA receptors function. Pay close attention, okay, to the digital mastery, which you're about to see. Ooh, look at that. Huh? Huh? Wow. That's pretty good. Yeah, that's good. That's good stuff. Yeah, good stuff. Okay. Thank you. Okay. We're not going to talk about mGlu receptors the rest of the semester, so just keep that in mind. They're not targets of psychotropic abused drugs that we're going to talk about. So there are no questions on exam 2 about mGlu receptors or metabotropic glutamate receptors. Okay? Now, know this: if I'm a neuron that has glutamic acid decarboxylase, do I produce glutamate? We just use glutamate as a substrate to make GABA. I told you don't need to know drugs, right? The FDA approved names of drugs used to treat seizure disorders. I'm just making sure. So I think whoever took it this morning, there was no question that they had to know one of these drugs that tried to change. That's fixable. It's fixable. Is that everything? Oh, we did acetylcholine too, didn't we? Where is it now? Why is it not on my? Yeah. Okay, let's talk about acetylcholine. Okay, other than choline, what's used to produce acetylcholine? Acetyl-CoA, right? Where do choline and acetyl-CoA come from? Food sources. Okay? Choline's not an amino acid, acetyl-CoA is not an amino acid. What's the enzyme that metabolizes acetylcholine after its release, has an effective neuron number 2? What breaks down acetylcholine in the synaptic cleft? Good, you're pronouncing that very well. Very good. What would happen to my mamaw with Alzheimer's disease if I gave her a drug that blocks acetylcholinesterase? Better memory. Okay. We just got it. Yes. So Said asked a question, why is that good? That's why I asked you, because that's an important question. So acetylcholine that's made in what part of the brain? Not made, synthesized. That's cell bodies. Basal forebrain, Open your mouth. Right here. Move your mouth. Those axons, a lot of those axons go to the hippocampus to release acetylcholine. That is a huge part of memory formation and long-term potentiation. So if we prevent acetylcholinesterase from breaking down acetylcholine, we get more acetylcholine release in the hippocampus, and I'm off to remember to turn off the stove. Those drugs don't work very well, but they're better than nothing. Which kind of acetylcholine receptor is ionotropic? What? Nicotinic. Nicotine. Named because nicotine binds it. Okay? Which acetylcholine receptor, when activated, produces a hallucinogenic effect that aided in the writing of one of your favorite childhood bedtime stories? Muscarinic. Yeah, muscarinic. Russians pronounce it muscarinic. Yeah, Mickey Mouse. Yeah. All right, I think we just covered acetylcholine. Specific questions? So you know the drill. Make sure you hit submit. Every question, submit for the exam. If you get kicked out during the exam, a Java pop-up, that's going to happen. I will have my proctor sheet. I will let you back in, unless it says you took a screenshot or did something dirty. Make sure you use Chrome. If you're using a tablet or a phone, download the Top Hat app. It's the only way you can take the exam on a tablet or a phone. Any questions? Okay. We'll see you Thursday.